So last lecture we all have seen that what are the changes which occurs after the death of the fish, that is the postmortem changes. In the postmortem changes we have learned that the first stage which occurs is the first death will occur, and why death is occurring, it is due to the suffocation. So it causes the hyperemia near the gills, and thereafter we have seen is the rigor mortis. In the rigor mortis, we have seen the pre-rigor, in-rigor, and post-rigor stage. So pre-rigor and the rigor, in-rigor, these two stages are the standard of the quality, or you can say are the quality index. So we are going to learn this particular stage, means the fish sailor, they have the criteria to maintain the fish into the rigor stage. So this rigor mortis is one of the stage when if it is maintained properly, so the fish quality is evaluated on the basis of it. So we are going to observe how the fish freshness. Now freshness means there are various phases of the freshness that we have already learned. So how can we evaluate that how old or how stale the fish is? So we will learn the quality of fish, and we have also learned once the fish when it leaves its rigor mortis stage, that particular time the chemical changes start occurring into the flesh of the fish. In the chemical changes, what we have learned, that first thing which is going to be happen, means after the post rigor stage, what is going to be happen, first the texture will change. That means the fish which was very stiff, now it will no more stiff, and its body will become very lean, and thereafter it will start producing changes into the taste. Now why the taste is changed, the taste of fish will change because of the chemical changes. Now what chemical change, first chemical change we have learned will be the autolysis. Autolysis means the changes or breakdown of protein into the amino acids. So it make you the bitter taste, foul smell to the fish. Then the next stage, when the fatty acid degradation will start. Why it happens? Because whatever oil is present in the body of the fish get oxidized, and this oxidation produces the rancidity. So these stages you must remember in a perfect sequence: hyperemia, then rigor mortis, then autolysis, then the rancidity. So let us begin. How can we evaluate? But before evaluating, what one must know, that what changes are going to be occur, what we are going to actually evaluate, right? So now those who are eating fish, they know very well that if the fish is identify, yes, the fish is identify, yes, so whatever fish is there, by its texture and odor only, if it is stinking, that means the fish is not proper. Now see, a peculiar smell is always there in the fish. Now you are going to the fish market and fish market is stinking, it does not mean that all fish are the stale one, because a peculiar smell is already always there in the body of the fish. So out of that, if some other foul smell, that means the oxidation smell or autolysis smell is coming, so that particular time the fish will be called as the stale. Now how can we assess the fish? What are the factors? Now see, with the help of the senses, what test we are doing is called as the organoleptic test. Organoleptic test means by proper observation, we are evaluating the fish, whether the fish is proper, or you can say that the fish is fresh or not. Now, most important thing, what we should observe whether the fish is fresh or not. First, the general appearance. Now, what general appearance? How the eyes are, then gills, then whether the surface is slime or not. What about the scale? What about the firmness? Whether the fish is firm or soft. Then odor. Now, thereafter, the appearance and the presence and absence of discoloration of the underside we can see. Then we can see the backbone. Then rigor mortis, absence of rigor mortis is one of the most important criteria. Now, for that you must know about the rigor mortis. In the rigor stage, what things should be there? If it is absent, it means it is called as the oily balls. Now, practical purpose for well-defined phases are there. Phase 1 is the first quality. First quality means it is the pre-rigor stage. Pre-rigor immediately after catch. What can we do? It is for fresh sale as well as for the freezing purpose. Then phase 2 is the second quality. Now, second quality means the fish has been taken to the icing or you can say the freezing plant, and their fish will be sold for the fresh and smoking and canning. Then the third stage is the quality for the smoking, canning, and the salting. Now, when they have to do the salting and all, so that particular time they are using the frozen fish. Okay. So that particular fish will be used. Then phase 4 is the last stage. Here we are going to use the condemned fish, means the fish which cannot be used for the purpose of the eating. Now, for what purpose then this fish is being used? It can be used for preparation of the fish meal. It can be used for the purpose of the fertilizer and other. So this way the fish can be evaluated. Let us see how can we do the organoleptic test. We are going to talk about the three types of tests: organoleptic test, then microbial test, then chemical test. So all these tests will discuss in this particular lecture. So first we will talk about the organoleptic. Now remember the term organoleptic. It means observation of the animal and testing, means evaluation by its general observation. Now, general observation means this particular test can be used for the small scale. Okay, it cannot be used for the large scale. A local fisherman selling the fish into the local market can use this particular test, but the big canning industry and all, they cannot rely on the organoleptic test only. Now, what are the general observation in the case of the organoleptic test? First, they observe the eyes. Now, when we talk about the eyes, the fresh fish, they will have the predominant normal looking and glistening eyes. The eye, you can see here the right mark is for the fresh fish. Now you will observe that the eyes are not sunk, right? It is proper. But when you will observe in the case of the fresh, if you will compare fresh with the stale, you will observe that the stale fish eyes are dull. Now here you can see the eyes are full and prominent, and it's looking absolutely normal like the fresh fish only. Then you will observe here that this fish, it has the sunken eye. The eyes are completely darkened. Yes. Then, moreover, that you will also observe that the eyes are not prominent and not at proper place. So, then next thing we can observe is the gill lamellae. This is a general test. When we go to the market, first we check the gills of the fish. So you will observe that the gills are proper. It's a bright, means you have to Open up the operculum to check the gills. Now you will observe in the fresh fish, gills appear very bright red and it will not look dull, gray or slimy. And when you will observe the smell, so the from the gill portion, the now see we know that the decay starts from the gills only, because the hyperemia is the condition which occurs at the gills first. So for that particular reason, the gill will start stinking first. So if the gills will have the normal bright red color, so that particular time the fish will have the normal fish peculiar smell. But at the time of the staleness, the fish gills will be darkened. It will be having the foul smell. It may also look like gray. And scales. Next thing is the scales. Now, if the fish is fresh, so that particular time the fresh fish will have the shiny scales, and the scales will not have any mucus. It will not be fallen type. And when you will observe the stale fish, you will find that the scales are fallen kind, and a particular type of the mucus is spreaded over there. So this is about the fish. Now, as I told you that we are learning this particular unit about the complete seafood. So next most important seafood is the prawns. So let us discuss about the prawns. When we talk about the prawns, in the case of the fresh prawn, you will find that they do have the proper texture, and their shelf life can be controlled up to the 8 days. Means 6 to 8 days they have the shelf life. They can control it, but thereafter what changes happens, that can be found by this normal observation. So one thing to be remembered, that they can be sold within the 8 days after the harvest. Now what changes occur into the appearance we will discuss. Most noticeable change is the progressive black discoloration of the head. Means black discoloration will start occurring on the head region, and as well as the tail region. Now why this is happening? Because of the melanosis. Melanosis is the process which will start occurring. Now next thing you will observe in the case that they have the fresh prawn, they will have the proper strips. Okay, proper strips means usually you will find white and yellow strips are there on their body, and here the distinct pattern will observe. Means you cannot clearly distinct bands will be observed on the body of this. The band will start disappearing, or you can say that they are just start decoloration. Now next most important criteria is the firmness. It is same as like the fish, that if the body is proper, firm, proper stiff, that means the prawns are the fresh. But if it is simply, now see, how can we check the stiffness of an animal? Just press it. So if you will press it, and that time it gives impression, that impression, now after pressing the with the finger, what will happen? Even if it is a fish, even it is a prawn, simply press the organism. So after pressing, if the animal is fresh, so the depression caused by your pressing will disappear. But if the depression remains, that means the body is not properly formed. If after observing, if you go to the market, you want to buy the fish, you want to buy the prawn, you want to check the stiffness. Stiffness can be observed very well by our senses. But even if you are confused whether it is stiff or not, simply press it. So after pressing, what is going to be happen? Your finger impression will come over there. And if this finger impression goes off, that means the fish is fresh. Understood? So this is the simple test. You can do it with your hand. Now see, in the case of the prawn, if the head is firm, so that particular, now here, see, this good one is the good. Head is firm, that means the prawn is fresh. But as the time goes away, so that particular time, the head of the prawn start becoming loose, and most of the time it just dislocate. And that, the most prominent region in the case of the prawn is the head, because lots of enzymatic and microbial degradation occurs. So that particular time it causes the changes within the 3-4 days, and it causes the discoloration into the flesh in the head as well as the collar meat. So discoloration of the head will occur. Then texture is one of the very good indicator in the case of the freshness. You will find that the gradual breakdown of the structure occurs. The peel will become loose, and the pH will also change. Now pH cannot be checked by the organoleptic test. Simply you have to do the microbial changes. But in the case of the chemical testing, the pH is checked in the case of the prawns. So this is all about the organoleptic test. So hope this will be very helpful for you, because whenever you go to the market, the fresh fishermen will definitely say, "Arey, ye taza hai, ye taza hai, ye taza hai." But no, until and unless you will not check it. Now, hope you understood what are the criteria for the fresh fish. What are the criteria? Body must be firm. This is one of the most important criteria. And I told you one of the tests, that it is a very simple test, that press it with your finger. If it is properly, depression goes away. Yes, definitely the fish or the prawn is fresh. Then second criteria I told you is what? The second most important criteria is checking the gills in the case of the fish, and checking the firmness of the head in the case of the prawn. Then third most important criteria in the fish, I forgot one criteria, that the vent region. In the case of the stale fish, it comes out. Stale, see, vent is the opening of the digestive system and urinary tract in the case of the fish. So if this vent region protrude out, it comes out, that means the fish is stale. But in the case of the fresh fish, the vent region is properly added. If you don't like to touch and your mother has sent you to buy the fish, just check that the external opening is proper or not. If it is coming out, that means the this particular fish is not fresh. So these are the certain criteria. Now see, only observation is not sufficient when we are going for the industrial level, because freshness is one of the most important quality criteria. So if the person is very experienced, they can judge the freshness of the batch of fish by just appearance, odor. But there are many situations when freshness must be expressed in some degree, right? Suppose if I'm selling it on the larger scale, I'm canning it. So that particular time, I need to express the freshness in some degree, right? So such as number, or it can be the scale, or it can be the grade. So it is very difficult to standardize the sensory method, since this is not possible to define or measure the freshness in the absolute sense. Now I can see, I have touched the fish, yes, it is proper, but it is not the standard method. So there are number of non-sensory methods also which can be standardized into the laboratory and based into the chemical and physical property of the fish freshness. Now all this particular further testing can be done on the basis of the physical and the chemical changes into the fishes. These tests do not measure the freshness directly as human can do. We can check the gills and all, but provide the major, means it will give this much particular count of so and so chemicals are there which accompany the spoilage and can confirm the score. It can give the grade which a sensory mechanism. Now see, organoleptic test is a sensory test, so that is insufficient. So what are non-sensory method? The first is the microbial method for evaluation. Now microbial method, why it is required? Now first of all, we must know that the why there is a need of the microbial testing, because the most important threat to the fish is the microbial spoilage. Now this is one of the very potent factor into the fish spoilage. It is not only completely destroy the nutritive value of the fish, but it also causes the poisoning in the person or the who is consuming that particular fish, or it can also cause the death of the person if the microbes are present into the fish. Now see, landed fish, it contains many microbes inside it, as well as virus, disease causing germs, which we are going to learn in detail, the fish diseases and the virus and the fungus which are causing. So immediately caught fish can also have all this virus. So Pseudomonas, out of that, the Pseudomonas we know are the bacteria which constitute more than 90% of the organism. Now, most abundant quantity of certain organism will be found into the decaying fish. Now, as far as living fish is concerned, so the number of the living fish, the number of microbes will be less. So the fish will become, because why it is happening that the living fish have the less number of microbes as compared to the dead fish. The reason is that the living fish have some type of resistance against the microbes. So for that particular reason, the number of microbes will be less. However, after the death, this resistance will ceases. So what will happen? Bacteria will become very rapid and it will cause the extensive spoilage to the fish. Now, high percentage of water content is also available in the fish, so it favors the microbes for bacterial multiplication, and bacteria may also infect the fish from outside during the careless handling. If the fish are not handled carefully, so that particular time the now see, this is about the microbes, that these microbes are present. Now these microbes are present by default in the fish. So this particular microbe will cause the harm. Now, if the fish is live, we know that the fish body has some resistance against the microbe. Active enzymes are produced, ATP is produced to fight against the microbes and all. So for that particular reason, the fish will not allow the rapid multiplication of the microbes. So what can we do now? How this information is helpful for microbial method of evaluating freshness? So we can do the total count. Now simply take a sample of the fish. Now observe. Now we can do the two things. Simply we can do the common plate count on the agar, as we are simply doing the air microflora practically in our laboratory. So same way, what can we do? We can take the small sample and just spread it on the agar plate and do the common plate count as we are doing the normal plating. Then thereafter, microscopic organism of the sample we can do. Then see, there are certain spoilage bacteria. So these bacteria or these microbe, they produce the certain type of the reaction. So if is, I have taken two example here. One is the Shonella putrefactions and another is the Viburnacea. So these two bacteria, they have the specialized spoilage reaction. Say example, Schwannella, Shonella, it can be identified whether it is present or not. About the spoilage bacteria. Spoilage bacteria, as I told you, that the Schwannella putrefacients, it can be seen as the black colonies. Now it can be seen as a black colonies. That means we are doing the plating of that bacteria. Now why it is causing? Because of the precipitation of the ferrous sulfate. Same way the another bacteria, Vibrio necia, it will also form the black colonies on iron agar. So that particular time, the presence of that particular bacteria, it can be detected, and this clear region of the bacteria. Now see, bacteria can be counted, as we all have done practical counting of the plate. Again we are going to do in this semester also. So this plating and this counting of the bacteria is very helpful for measuring that how many harmful bacteria or how many colonies are there. Then there are various chemical testing. Now see, chemical testing is done on the basis of the chemical changes into the spoiling fish. Now see what happens. The chemical composition of the fish, or you can say the texture, along with the texture, the chemical composition also changes, as we all have learned the autolysis and the rancidity. So it brings the changes into the odor, texture, appearance, as well as the flavor. So spoilage is the result of many complex changes, and most of the chemical method involved into the measurement of one or many substances. So no chemical method will give the same degree of accuracy. All the methods are different. So first method is the trimethylamine method. Trimethylamine test is the most important test for the freshness. Now see, this can be efficiently used to the cod fish, then haddock fish. In the normal or the fresh fish, trimethylamine oxide is one of the most important component of the fresh fish. Now what happens? This trimethylamine breaks down by the bacteria, and this bacteria gives out the odoriferous substance which is known as the trimethylamine. Related chemically to ammonia, means is just think like ammonia. So what can we do? We can count how much trimethylamine is produced. So consequently, we know that the fresh fish will have the very low quantity of the trimethylamine as compared to the stale fish. So the spoilage of the fish will raise the concentration of the trimethylamine. I repeat, what happens in the normal fish, TMA or trimethylamine value will be less, and that why it is less? Because it is present in the form of trimethylamine oxide. So its oxides will be break down by the bacteria. Why bacteria will break down? Because we know as the fish goes stale, so that particular time the amount of the bacteria increases. If the number of bacteria will increase, definitely it will do the more and more chemical reaction. So this TMA value in the case of the stale fish will increase. So we can evaluate it with the fresh fish. Then second, now this will give the proper number. This much TMA is been increased, right? Then total volatile basis, there are several other similar substances produced during the spoilage. This group of substance, rather one is estimated here, true only for the later stages of this. Now see, total volatile basis mean, first we will check the ammonia, whatever substances are produced. So at the total volatile substances, so total volatile basis comprised of the ammonia, hypoxanthine and all. So that particular time, what happens? We check that amount or the chemical which is present in the normal fish, it is varying from the stale fish. So we just do the quantitative checkup. So first we'll talk about the ammonia. Ammonia is produced by the bacterial breakdown of the muscle protein, and it appears in the quantity at the relative late stage of the spoilage, and the test will give indication only of how poor sample is. Now, if the sample is poor, so what will happen? That the ammonia will be found in the all flesh part. Then next is the hypoxanthine. Hypoxanthine is the normal constituent of the fish flesh, but present in a very, very low concentration in the living animal. And after the death, balance of the enzymatic reaction disturbs, which keep it low and distributed. And because of that, the content of the hypoxanthine increases. So this is about the uh, chemical tests. So three type of tests we have learned for the evaluating the freshness. The first organoleptic test. In the organoleptic test, we all have learned that by appearance, how can we find whether the fish is fresh or it is the stale. Then in that we have kept certain criteria in our mind: stiffness, then the gill checkup, then in the case of the fish, we will observe the eye, gills, vent. Okay, in the case of the prawn, simply check its head and the pill. If it is firmly attached, yes, the specimen is fresh. Then next we will have learned is the microbial count. So we know that the microbial count will increase, and at the end we have learned the chemical methods. In that two methods, that is the trimethylamine and volatile bases. Two volatile bases we have learned: ammonia and hypoxanthine, both increases into the stale fish. So this is all about for the chemical method of the evaluating freshness. Now, after that, this is all about the quality of the fish. Now, what is the name of our unit? Quality control and packaging. So now, see, we have controlled the quality first. We have understood what are the criteria which should be maintained into, or you can say what quality we require. Okay. So first we discuss about quality. So this is the quality part. So here we end up with the quality part. So what is the fresh fish and what are the changes occur into the body of the fish and how can we evaluate is clear. Now thereafter we will learn and we will go towards the packaging, because finally this particular unit or this particular subject we are learning as an applied component. We are going to use it for the economic purpose, right? So now we have evaluated the quality, how to take the product in the market. So for taking the product in the market, we need to make it the presentable. But before, this is about the assessment of the quality part. Next, when we are taking it to the industrial level, we will first take care of the sanitary. So next lecture, we will talk about the sanitary operation. So for this note for today, I like to stop now. Dear students, hope you all have watched the complete video lecture, but still after watching the complete lecture, if you have any doubts, don't hesitate to ask me your doubt. You can write your doubts into the comment box, and if you want more video updates, definitely you can subscribe to the channel. Thank you for watching. Watch the videos with a proper care. Make sure that you are noting all the important points, and at the time of the exam definitely these notes are going to help you. Thank you.